Good evening, everyone. Thanks for coming. So my name is Raquel Vasconcelos. I'm teaching the psychology course, and I'm here to present George Augustine. First of all, I want to say that, as you probably know, we are going to have a neuroscience laboratory here at University of St. Joseph. And regarding the research lines we want to approach, we will particularly focus on behavioral plasticity, on hormonal control, and also on sensory perception and encoding in the brain. So, besides this, we are quite motivated to embrace other areas of interest, such as arts, architecture, and business. So, in a perspective that could be like a translational research, like establish bridges between these several areas. So, in this vein, I invited Professor George Augustine. I had the wonderful chance to meet him during my research at the Marine Biological Laboratory in Woodsall in the States, Massachusetts, and while I was doing research during my fellowship there. So Professor George Augustine, he did his PhD at University of Maryland in 1980, actually when I was born. I don't know if this is of interest or not, but... And he actually did his PhD on a very interesting topic. So he was investigating a substance that is toxic, and it was present in, the chocolates, M&Ms, right? Right, yes, exactly. There are actually some that are red, and so within that pigment there was some kind of a toxic substance, and he was working on that. So it was really good for consumers that he discovered that this was bad for our health. Okay, so besides this, afterwards he did his postdoc at UCLA University in California, and he started to become very interested in neuroscience research. So he used a model which is the giant squid. They have huge neurons, they have remarkable neurons, so they are perfect for research. So he was looking into the synaptic transmission in this model species. And he also did fantastic sabbatical research in Germany with a Nobel laureate, Erwin Neuer. And Neuer, he was actually the inventor of the patch clamp technique. I don't know if you are probably not so familiar with this technique, but it's really remarkable for electrophysiology research. In the meantime, when he finished this sabbatical, he had a teaching position at Duke University Medical Center, and he's right now presently the director of the Center for Functional Connectomics in Korea, and he's also a professor at the Duke NUS Medical School in Singapore. So his present work basically focuses on synaptic transmission and understanding the neural circuits within the brain, which is really important. So please welcome Professor George Augustine.
Thanks very much, Raquel. Can everybody hear me? I'm not going to try too hard to speak into the microphone. Usually my kids complain that I'm too loud, but if you can hear me, let me know and I'll use the microphone. Thanks for the great introduction. Anytime you need to be introduced, ask Raquel. She's spent all afternoon working on my introduction. She's really the most conscientious introducer I've ever seen. So, yeah. Seriously, it's really exciting that the university is embarking on a new neuroscience program, a new neuroscience laboratory, and I think in Raquel you really have one of the very best here on your faculty. So I'm very excited to be here. This is my first time in Macao, and it's great to be able to meet so many people and learn so many things today. Thank you for inviting me. Before I get started, I encourage you to stop me anytime if you have any questions or comments. No need to wait until the end or to the reception afterwards. I'd like this to be as interactive and as bi-directional as possible. So please don't be bashful about asking questions. As Raquel mentioned, what we're going to talk about today is the brain. And in my mind, as you can see here, I consider this to be really kind of the last frontier of human intellection. I mean, there's lots of things that we don't know, but I think in terms of fundamental challenges, important things that we know almost nothing about, I think the brain has to win hands down. And so that's what I'm going to talk about today. I'm going to talk about not so much a finished story, but a concept, a technology, a way to understand the brain. And so why haven't we been able to understand the brain? People have been studying it for more than 100 years, but why is it that we don't really know how it works? The problem is it's very complex. Within the brain there are many nerve cells, neurons, that seem to do most of the computational work. There's more than 100 billion of those. And it's not even one kind of nerve cell. There's many different kinds. So there's a tremendous cellular diversity. A lot of cells, a lot of different kinds of cells. Makes the problem difficult. Even worse, it's not the neurons per se that are really responsible for the information processing, but rather it's the wiring between the neurons. The neurons form synaptic connections with each other, and to the tune of approximately 1,000 synapses per neuron. So if you multiply this huge number, 100 billion, by 1,000, you get an even huger number, 100 trillion. So there are more than 100 trillion synaptic connections, and it's these synaptic connections that are really literally the wiring of the brain. So they're responsible for information processing, they're responsible for information storage during learning and memory. So and if we want to understand the brain, what we need to do is to understand what all of these connections are and how they work. So let me introduce a little bit of terminology. I know some of you are, maybe most of you are not from the neuroscience field, so I'm going to try and take it easy on the jargon, but let me introduce this concept to you, this word called the connectome. So the connectome is all of these 100 trillion synaptic connections. This is the wiring of the brain. So we can say that if we want to understand the brain, what we really need to do is to understand the function of this connectome. And so why haven't we been able to do that? Well, it's huge. Studying 100 trillion of anything is difficult. Okay, so historically, the way that people have tried to get a handle on this problem is through the use of anatomical techniques. So with anatomical techniques, we can see the individual nerve cells in the brain, the neurons, and we can imagine their synaptic connections. So here's an example from the classical work of the most important, most influential neuroanatomist of all time, Santiago Ramón y Cajal from Spain. And what you can see here, he made many, many thousands of these kind of diagrams, but you can see our individual nerve cells, and you can see that they send their processes up, and they seem to make connections with other nerve cells. So that's the measurable. That's what he could actually observe. And from these observations and his keen instinct, he was able to deduce that information should flow in the direction shown by these arrows. So going from this nerve cell to this nerve cell to this nerve cell and then out again. As it turns out, he was right about this one. But it was, I would say, more luck than anything else, because anatomy can't really tell us about function. It doesn't tell us for sure whether this really is a synaptic connection between this nerve cell and this one. And even if we could conclude that it was, we couldn't know whether it was strong or weak or fast or slow or excitatory or inhibitory. So we really can't deduce function from structure. So in order to understand the function of this absolutely minimal circuit just between two nerve cells, what we need to do is to this kind of simple experiment. So what we need to do is to be able to stimulate the neuron that's doing the communicating, the presynaptic neuron, in this case this structure called a parallel fiber. And then we need to be able to detect or record a response in this receiving neuron, the postsynaptic cell. So if we can stimulate this one and see a response in that one, then we know that there's a functional circuit, and we can even learn a lot about what the precise function is. So reducing this complex problem, 100 trillion connections, down to its simplest, most fundamental unit, what we need to be able to do is to stimulate one neuron and record from another. Sounds easy, right? Well, as it turns out, for technical reasons, it's very difficult to do this. And in the interest of time and the interest of trying to keep things as non-technical as possible, I won't go into all the details, but it's hard. So that's why very few people are doing this successfully. So what I'd like to tell you about today is a new kind of technology, which I think really offers practical hope of doing this kind of experiment for all 100 trillion connections in a brain. And this is based on the technology called optogenetics that Raquel mentioned to you briefly. And so the approach that I'm going to propose to you today we call optogenetic circuit breaking. So here's our mission statement. Use genetically encoded optical probes to define functional, and that's the key word, functional neural circuitry. So let me parse that out into the three important terms. Genetic. I told you the problem is there are too many kinds of cells in the brain. Well, the genetics offers a way to get around that. There are molecular genetic tricks that allows us to interrogate one kind of nerve cell at a time. So we can achieve cellular specificity. So genetics, good. Optical, this word here, is the next important part. One of the technical problems with conventional techniques is that when you want to look at the function of neurons or their synapses, you can usually look only at one at a time. So if we have 100 billion neurons and 100 trillion synapses, you can do the math. It's going to take forever and a day. It's going to take thousands of years, even if everybody in the whole world was doing this kind of experiment, which of course they're not. Okay, so optical is a way around this problem. By using light, instead of looking at just one nerve cell at a time, we can look at a large number of them. And even better, one of the most important ways that information is processed and stored in the brain is spatial. So the circuits between nerve cells are organized often in very spatially precise ways, which can only be appreciated using optical approaches. So, and I already told you the importance of function. It's not good enough to just see structure. We need to know what the structures are doing. Okay, so that's our mission statement. And so combining these two words, optical and genetic, leads to this term, optogenetic, which is this technology which I think is fair to say is revolutionizing neuroscience. And what I'd like to do today is tell you about it. So for the purposes of mapping out the connectome, the functional connectome of the brain, we can restate our fundamental problem in optogenetic terms. So what we need to do is to have an optogenetic way to control the activity of the talking neurons, the presynaptic neurons, and we need to have another way to optogenetically monitor or measure the responses of the receiving neurons, the postsynaptic neurons. So if we have such a tool, and we have such a tool, then we should be able to do this kind of thing. Is it clear? Can everybody hear me? No? Okay, good. All right, so what I'd like to do today is to give you a brief progress report on how the technology is coming along for doing this and for doing this. Thank you for coming. It's nice to have such a young and non-biased member of the audience here today. Okay, so first let's talk about optogenetic ways to control the activity of the presynaptic neurons, the talking neurons. And the first and probably most important new development that I want to tell you about is a way to use light to activate neurons, photostimulation. And it's based on a very special kind of protein called Channelrhodopsin 2. So Channelrhodopsin 2 is not normally found in our brains. It's usually found in algae, Chlamydomonas, a single-celled aquatic algae that uses this channel to know where the sunlight is and move toward the sunlight. But what you can do is take this gene out of the algae and transplant it, again using molecular genetic tricks that I won't tell you about, into different populations of nerve cells in the brain. And so what happens is that once you express this, as it's called, once you transplant it into the neurons in the brain, this is what happens. If you shine light on the brain, only the neurons where you've transplanted this Channelrhodopsin 2 thing will be activated. So what happens is that they absorb light of a certain color, blue in this case, and what that does is cause the protein to change its structure so that it allows fluxes of cations, like sodium and potassium, to flow across the membrane of the neuron. And you can see that on the next slide here. Oops, this was supposed to be gone. Where we've taken a neuron from the brain of a mouse, where we've transplanted the channelrhodopsin into a certain kind of neuron called the pyramidal cell in the cortex, the cerebral cortex, the part of the brain that's important for kind of higher level processing and cognitive functions. And so ordinarily, if you look at one of these neurons and shine light on it, let's say we turn on the light here and turn off the light here, nothing happens. That's because almost no nerve cells in our entire body are sensitive to light. The main exception, of course, and the most important exception, are the photoreceptors in your eyes, right? We have to be able to detect light to see. But other than that, the rest of the brain is pretty much oblivious to light, as you can see here. Nothing's happening. However, in the neurons where we've transplanted the channelrhodopsin, when we turn on the light, we can see this flux of the sodium and potassium that I was telling you about. So there's an electrical current generated, and you can see its properties here. Okay, and this is a huge current, and we can tell how huge it is by looking at its effect on the electrical activity, the firing properties of these nerve cells. And so that's shown here. So starting here and ending here, we shine light on the nerve cell and look at its membrane potential, its nerve impulses. You can see if we shine a very dim light, we see just a tiny response. You probably can't even see it from the back of the room. But as the light gets brighter and brighter and brighter, you can see that the nerve cell gets more and more and more active. We've made it fire. It's magic. Normally they don't care about light. Suddenly they become very sensitive to light. So we can very precisely control the activity of the nerve cell according to the amount of light that we shine on it. Okay. Any questions? Oh, cell phone. Okay. Yeah, so it's really remarkable. So out of all the nerve cells in the brain, only this one kind responds to the light. And by using different genetic targeting strategies, we can make a different kind of nerve cell or a different kind of nerve cell sensitive to light. Okay.
You said you transplant anything to the neurons? Transplant?
Yes, I'm using that term kind of loosely. So what we do is make transgenic mice. So we take the DNA out of the algae and then force it into the cells that will become the nerve cells in the brain of the mouse. That's how we do it. Yeah, I can give you a lot more detail if you want, but that's the basic idea. So we use molecular genetics. So the DNA encoding the channel is told to go to a certain kind of neuron in the brain. If you're asking that. Good question, and the first question, maybe other people will feel more comfortable in asking questions. Okay, so how can we use this to map circuit activity? So one way to do that is actually something like shown here. Instead of this being a red laser, if this was a blue laser, this would allow us to map the connections, the circuits in which this neuron participates. So here's an example of such an experiment. So what we do is take the brain out of one of these mice that has the channelrhodopsin transplanted into a certain kind of neuron. We can do an experiment like this. So the neurons in green have the channelrhodopsin in it, so they're sensitive to light. And the neuron in red does not have the channelrhodopsin in it, but it receives synaptic information from the green cells. So we can use electrophysiological techniques like the patch clamp technique that Raquel mentioned during her introduction to look at the electrical activity of this nerve cell. And then what we can do is scan the laser, the blue laser spot, kind of systematically across the brain tissue, like this. And so the idea is that whenever the laser spot encounters a neuron that has the channelrhodopsin in it, it will fire an action potential, a nervous impulse. And if that neuron is talking to this neuron, then we'll see a response, like this. Conversely, if we shine the light on another neuron, like this one or this one, these gray ones, that don't talk to the red neuron, then we won't see a response like that. And so we have a kind of a robotic microscope that will do that kind of experiment automatically for us. It scans the laser spot across the specimen, and it also measures the response in the red neuron. And so what it can do is make a map that's shown here. So here's what the red neuron looks like in real life. And these pixels here show the position of the laser spot, and the color scale indicates the size of the response in the red neuron. So when there's nothing, it's blue down here. So you can see most of the places there was no response. But you can see that when we get close to the red nerve cell, there's responses. The hotter the color, the larger the response. Okay, so this corresponds exactly to that, and this interpretive diagram shows us what this map of the synaptic inputs means in terms of circuitry. So it shows us that right around the red nerve cell, there are a bunch of these green nerve cells that are innervating it and are exciting it, and whereas the gray ones are not. So that's connectivity. That's the connectome of this particular red nerve cell. Okay, so that's nice, but what's so exciting about this? The exciting thing about it is that it takes less time to do this experiment than it took me to describe it to you. We can do this experiment in 2 minutes. And using the conventional kind of technologies that I didn't go into in great detail, it would have taken approximately 1 year, and the data would not be as good as this. So you can see it's much more efficient. In engineering terms, there's a huge improvement in throughput, about 10 to the 4th or 10 to the 5th, 4 or 5 orders of magnitude. I'm sure all of us would be happy if our salaries suddenly increased by 4 or 5 orders of magnitude. And really, honestly, this is equally exciting to me, probably more so, because I know this one can happen. Okay, so that's what we can do with this kind of thing. That's one example. Any questions so far? Okay, yeah.
In this situation, we need to use this technology.
Okay, right now we're talking about mice, and a little bit later I'll tell you about worms. It's a little lower life form. In principle, it could be done in humans. Now, there are ethical issues, and there's 2 kinds of ethical issues. The first kind of ethical issue is this is a gene, an algae. So in order to transplant it into the human brain, you have to do gene therapy. And there are a lot of physicians and a lot of scientists who are very nervous about gene therapy, in part because some of the earliest trials of gene therapy in the United States killed the patients. So we have to be very careful about that. And the technology for doing gene therapy has gotten a lot better, but still, people are going very slowly on that. So that's one kind of ethical issue. The other kind of ethical issue, which is a little more of a philosophical one, which is, do you want to have the ability to control your neighbor's brain? And this is a kind of a profound issue. I'm not saying it's right, I'm not saying it's wrong, but these are the kind of things that people are worried about. I'll talk a little bit more about translational applications of this technology, including the use of human brain. And there, in fact, are companies that are spinning off to do this kind of stuff. So it's going to happen sooner or later. But for sure, right now we're concentrating on animal models because there's a lot to learn there, and also it's much better to test out the technology on animals compared to your neighbor. Great question. More questions? Yeah.
How did you figure out that the response here is in the brain and not in the algae?
Well, it's both, but there's no algae under our microscope. We only have this ion channel, this protein that we took the DNA from the algae, put it into the nerve cells, and then they grow up this protein. So we know that it's coming from the nerve cells. In fact, we know precisely which nerve cells it's coming from. That's the key information. No questions? You have a question there? I know you guys are going to ask questions. Okay.
Is this a destructive test? Do you have to sort of take the brain and then slice it?
Yes.
And shine the light on it?
Another excellent question. Yes, this is a very kind of reductionist thing. So what we've done is taken the brain out of the mice, chopped it up into slices, and then we put one of the slices under the microscope. But the next thing I'll show you, perfect segue into the next slide, I'll show you that we can do exactly the same thing in the living and behaving mouse too. No chopping up the brain required. Great. I'm glad the questions are starting. Please keep them coming. Okay, so as you heard from the last question, it's important to think about whether we can only do it in isolated brain tissue or whether we can do it in the whole organism as well. And this is such an experiment. So what we've done is shown here. We're looking down on the top of the skull of the mouse. What we've done is peeled back the skin of the mouse over the skull, and then we made the skull transparent. We treat it with a special kind of Japanese dental acrylic that makes the bone on the skull transparent, so we can shine blue light through it. Those of you who've seen brains before, you can recognize here's the brain and here's the blood vessels, and this is in the living, breathing mouse. Okay, so if you look over here now, here's the experiment. This is the again, this is for the scientists. This is the objective lens. This is delivering the little flashes of blue light at different spots, like this one, onto the skull and onto the brain of the mouse. And then the mouse is clamped under the microscope, and he's lightly anesthetized. He's not completely asleep, but he's mostly asleep. And so what we're going to do is shine the spot of blue light onto the brain of this mouse, and this mouse has the channelrhodopsin, the light-sensitive channel, expressed in the motor cortex, a part of the brain that's responsible for controlling movements. And I'll show you this little movie here that shows what happens when we shine the blue light onto the brain of the mouse. Yep, I'll show it again. One more time. Hello, Macao. It's waving to you. Okay, so it works in the whole mouse. And we can measure, you can see in that case, the right forelimb, the paw, was moving. That's why it was waving. And we can measure those movements. I won't tell you how, but trust me, we can do that pretty nicely. And then we can do the same kind of mapping experiment where we scan the laser spot over the right part of the brain and the left part of the brain, and then measure how much the right paw moves or how much the left paw moves. And then we can again use the pseudocolor scale to map the amplitude of the response. The hotter the color, the bigger the movement of the limbs. And you can see if we shine the light on the right part of the motor cortex, the left hind limb moves. If we shine the light on the left part of the brain, the right hind limb moves. You can see that these are aligned exactly the same. You can see this is on the left, this is on the right. And so we can make these maps. And we can make different limbs move, we can make the tail move, we can make the whiskers move, etc. So what we can do is look at the location of the laser spot and then identify the nerve cells that are responsible for controlling movement in these different parts of the brain, the motor cortex. And in fact, a fellow named Penfield did this maybe 80 years ago in humans, and probably most of you have seen in some way or another a depiction of his work, this so-called homunculus, where he opened up the skull of his patients and then micro-stimulated in different places and could see, the hands move or the lips move or the tongue move, etc. And so we can do the same thing in the mouse, but non-invasively. We don't have to open up the skull of a mouse to do that. So it can be used in the whole mouse, the whole animal, in addition to the extracted pieces of brain tissue. So hope that answers your question. Okay, any questions about the moving mouse experiment? Okay, so I'll summarize. There's too many words on here. I told you about this special kind of light-sensitive channel from algae, Channelrhodopsin, and we can use this to control the activity of genetically defined types of nerve cells, and we can use that to map the connections between different kinds of nerve cells, both in the isolated brain tissue and in the whole mouse. And this allows us to determine whether neural activity is sufficient for the circuit behavior or for the animals' behavior, right? If you can turn on the neuron and see a response, then you know that that activity in that neuron is sufficient to cause the response. Okay, so about translational stuff, what can you do practically with this stuff besides figure out the wiring of the brain? Here's a couple of ideas that people have been bandying about, and I emphasize this is a new technology. I think, we can do so many things that we couldn't do before that really it's limited by your imagination. So this is not an exhaustive list by any means. So one thing that a lot of these new companies are starting up to do is called deep brain stimulation. Some of you may know about this. This is clinically a very important therapy for movement disorders like Parkinson's disease, and is starting to be used for psychiatric disorders like depression and other kinds of things. So what you do is put an electrode down into some part of the brain and then pass electrical current through it to activate the neurons near the electrode. And it works. It's sometimes remarkable, the improvement. I just saw a movie recently where patients in a coma were given deep brain stimulation, and they wake up and are perfectly normal. It's incredible. It's like magic. The problem is you don't know which nerve cells you're stimulating, so it's possible that you're not stimulating the right ones, or you're stimulating some too much or not enough. And so by using photostimulation with channelrhodopsin, we can achieve a much higher degree of specificity. And so that's something that I think will happen, again, in due course, but we have to work out the issues that we talked about before. Another thing that I think will be useful in the future is this so-called brain-machine interface. This is something that's getting more and more exciting all the time. So the basic idea is that what you can do is use electrodes to measure brain activity, and you can use that brain activity to control a prosthetic, like a robotic arm and so on. Very exciting. It's, for people who are paralyzed, this offers tremendous hope. It's really a big thing. But one of the limitations is getting the feedback. Once, for example, the robotic arm moves, how do you know how much it's moved? Do you know if it's moved enough? I mean, for gross movements, your visual system can tell you. You can see the arm move, so you know it moves. But more subtle movements, or if it's a matter of touch, or sensing temperature, or who knows what kind of stimuli one would like to know about. But anyway, you can put sensors on the prosthetic device to measure these things, but then you have to feed it back, that information, to the brain. And so channelrhodopsin could be very useful for that, just in the way that you saw the spots of light moving the limbs. You can use that to provide information in real time about sensory experiences by these prosthetics. Last but not least, as I said, what we do with the channelrhodopsin is turn on the activity of neurons, and I think this is going to be a very useful way for screening drugs that are useful for channelopathy, so neurological diseases that are due to defects in these ion channels that are responsible for making neuronal activity. Last but not least, let me just briefly tell you about a couple of the people who are involved in this work. Hard to know where to start. Let's see, start with this guy, Guoping Feng. He's our mouse molecular genetics collaborator. He's been very helpful for a number of years. Let's see, the data that I showed you today was done by Masanori Matsuzaki, mainly. I should also mention my former student, Karl Deisseroth, who's a faculty member at Stanford University in the US, and he's the genius who first had the idea of using channelrhodopsin for photostimulating neurons. Question?
If I understand you correctly, the key advance here was to genetically alter the brain cells with this light-sensitive
Confer light
DNA.
sensitivity. Yes.
Now, that doesn't mean that you can actually distinguish varieties of neurons, right? You still,
Yes, we can
basically
the
have to turn on all of the kind of neurons, correct?
Well, you can turn on all of one kind. Depends.
Yeah.
Yes, that's right. And again, I didn't go into any technical detail. I'm happy to talk to you about it later. But yes, they're by using different kinds of cell-specific promoters, it's called. You can specifically target that channel to this kind of neuron, this kind of neuron, or this kind of neuron exclusively. So out of the thousands of different kinds of neurons in the brain, we know that even if we shine lights on the whole brain, only that one kind of nerve cell can respond.
And is the pattern of patterns significantly different from different neurons?
Oh yes, in terms of their circuit response, their behavior response. Sure, every kind of nerve cell has a different function, definitely. Yeah. I could have given a more detailed version of the seminar where I could prove that to you, but definitely, there's a reason that there are these many different kinds of nerve cells that all have different jobs.
But I'm interested here in the what is the varieties and patterns of signaling?
Well,
What does that tell you?
let's say, well, I mean, in this paradigm, it's kind of all the same. Every time we shine the flash of blue light onto the nerve cell, it fires the impulse.
Right.
That's what we want. So at that level, they're all the same.
Right.
But what differs is which nerve cells that one's talking to. Those nerve cells are different in each kind of nerve cell.
And it all reinforces the kind of mapping that people have already been doing of the brain.
That's right. It certainly doesn't contradict anything that we knew from the academy, but what it does is enrich it in several dimensions.
It makes the analysis better.
And makes the analysis function. Yeah, huge. Questions? Yeah.
Years ago, I attended a conference by Professor Damasio.
Tony Damasio. Yes,
Damasio,
yeah.
yeah. And it was about the good and the bad inside our head. And he spoke about this lady from Baden-Meinhof, the group.
Yes,
and she has first she was a very quiet and calm person, and she had an accident. And she broke her head, and then she became very, after that, a terrorist. And she only thinks about killing. So this is something that can prove that we have bad neurons and good neurons.
Yes, sure. Well,
Or
there's a
when
lot
you
of
stimulate,
complicated stuff going on.
yeah, you can stimulate good and you can stimulate bad.
In
The
principle.
question,
Yes.
the question before it was how can you distinguish what you're doing?
Right.
You have the answer, okay?
Right. That's right. That's exactly right.
And but you can use all this knowledge for good and for bad.
Well,
And
that gets
you can
into this
influence-
ethical point that we were talking about before. At the level of the mouse, it's all good. It's all information that we can use to understand our own brains better. But yes, when and if we get to the point where we're using this, maybe a lot of people aren't thinking about that, then this ethical thing really comes to the fore. Yeah, but that's true for any technology. Anything powerful can have good outcomes or bad outcomes, and I think that's why we have committees to determine the ethical use of these kind of technologies, especially when it comes to humans. Good point.
There are so many kinds of different neurons. Are they responding to one specific type of neurons that responds to a specific type of wavelength? Or certain type of neurons respond to certain
With this particular channel, it's pretty much only sensitive to one color of light, blue. We now have other ones that respond to red and yellow. We have other ones. So what we can do with that is we can put the blue one into one kind of neuron, and the yellow one into another kind of neuron, and the red one into a third kind of neuron.
How many colors are used to differentiate different kinds of neurons? There are so many neurons. How many colors?
That's a good question. Ultimately, that depends on how well tuned the channel is to one particular color. So if you have a channel that really only cares about blue light, then you could have many channels. If you have a channel that's sensitive to blue light, but it also kind of spills over into green and yellow, then that's going to limit the number of your channels, the bandwidth, as we call it. So far, unfortunately, these channels have kind of broad bandwidth, although I'll show you an example where we can get two channels pretty easily. Two channels of information with different colors of light. But one of the things that we want to improve in the future is tightening up the bandwidth of a given channel so that we could get more colors.
Right?
Yeah.
Internet talking.
Yeah, it's funny, when we had no channels, we were really happy with one. But once we get one, we want two. Once we have two, we want four.
Building up of the channel.
Yeah, that's right. It has to do with so far, we've mostly used what Mother Nature has given us. As I say, we just took this thing out of the algae and it miraculously works in the brain of the mouse. But yeah, in the future, what we're going to do is spend more time engineering the channel to optimize its properties, in part in the way that you very astutely. Yeah.
Okay, how can you put light inside? Do you need a device to go?
Let's see. Well,
internally, or can you use outside lights?
Yeah. Well, for the experiments that I showed you with the waving mouse, that was all external light. Right? We're just shining light down on the skull. But that only works because of the light scattering properties of the brain. That only will get us down maybe 1 millimeter into the brain tissue. So the rest of the brain is dark. But what we can do then is put optical fibers to go as deep as we want to do. Now that's, I don't prefer that way because it's a little bit invasive, but honestly compared to, deep brain stimulation where you put an electrode down into the brain tissue, it's no worse.
I can understand how this works when you're looking at physiological response. You shine a light and the arm moves, whatever.
Right.
But that doesn't tell you anything about mental processing.
Well, as I said, it can tell you a certain kind of thing, whether the activity in that particular kind of nerve cell is sufficient. So if you turn on those neurons and you see whatever, this,
some sort of physical
something, then you can conclude that those neurons are sufficient. The activity of those neurons is sufficient. What it doesn't tell you is whether it's necessary, right? It could be that stimulating any kind of neuron in the brain could make the pop. That's not in fact the case, but in principle it could be like that. And what I'd like to tell you about next, again, your question is spot on. The next thing I want to tell you about is a different kind of optogenetic technique that will allow us to look at whether some activity in a certain kind of nerve cell is necessary. So instead of turning on the nerve cells, you have to be able to turn them off. We can do that too. Any more questions? No. Okay, great. Let's press on with that. And so, so what we want to do to do the experiment that he proposed is to be able to turn off nerve cells with light photoinhibition. So the opposite, the complement, logical complement to what I just told you about photostimulation. And fortunately, we have tools to do that now, like this thing called Halohodopsin. So Halohodopsin is from a different microorganism. It's from a bacterium that responds to light. And what that thing does, and again I'm leaving out all the technical details, is it's sensitive to a different color of light, yellow. And when this thing sees yellow light, what it does is pump, instead of being an ion channel, it's an ion pump. It pumps a negatively charged anion called chloride into the nerve cell. And what that does is shut off the nerve cell activity. And again, I'd like to emphasize, because we'll come back to it in a minute, it's responsive to a different color of light, yellow light. Remember, Channelrhodopsin likes blue. This one responds to yellow. So again, we can use molecular genetic techniques to transplant this Halohodopsin into different genetically defined populations of nerve cells. And sure enough, when we shine the yellow light onto the nerve cells, you can see this flux of chloride ions. This is showing it electrically. And maybe a better way to look at it is here, where the nerve cell is firing impulses, and then we shine the yellow light on it, and boom, it quiets down. And as soon as we turn the light off, it starts firing again. So it's a way to rapidly and reversibly silence neurons so that they aren't active. Okay, and as I said, Halohodopsin is sensitive to yellow light. So in principle and in practice, as you'll see in the next two slides, you can use one color of light to turn on the nerve cells and another color of light to turn them off. And here's such an experiment. So this is a nerve cell that's expressing both channelrhodopsin and halohodopsin. So first we shine the blue light to photostimulate the neuron via the channelrhodopsin. So you can see, sure enough, every time we shine the light, it fires a nerve impulse. And now we shine the yellow light to activate the halohodopsin, this chloride pump. And now you can see that when we shine the bars of blue light, no impulse. There's only a sub-threshold, small response here. Turn off the yellow light. Sure enough, it comes right back again. So you can do both. Have bi-directional control. We can turn them on, turn them off in the same nerve cell. And here's a behavioral example, not in a human, not even in a mouse. This is in a worm called C. elegans. Why do we care about worms? Because genetically they're one of the most manipulable animals. Sidney Brenner won the Nobel Prize about 10 years ago for realizing that this worm is very good for genetic studies of the nervous system. And now worms, worms aren't going to be giving you lectures anytime soon. Their intellectual repertoire is pretty limited. So when you want to study worm behavior, you look at things like their movement. Okay? And usually they kind of wiggle around in a sinusoidal wave, and you can measure that. And then so what these guys did, sorry you can't see it down here, that's an experiment from Karl Deisseroth's lab, my former student that I told you about. So what he did was put channelrhodopsin into the neurons or into the muscle cells that are responsible for contracting the worm and making it move. And sure enough, if you shine the blue light on the worm, he shortens, the worm contracts. Now if you also shine the yellow light, this is a complicated figure, but just compare, let's say, this red bar to this red bar. What you can see is that the yellow light causes the worm to relax. It gets longer again. You turn off the yellow light here, and then he contracts up again. So you can control the behavior of the worm bidirectionally. The blue light causes it to shorten because it's excited, and the yellow light causes it to relax because it reverses the excitation. So you can have bidirectional control not only of neuronal activity, but of the behavior of worms. And again, this should be applicable to mice, and eventually to humans. Nobody's doing humans yet, but I think monkeys are pretty far along, actually. Okay, so to summarize this, so I told you about this other kind of optogenetic tool, Halohodopsin, that allows us to photoinhibit, so to control the activity of neurons in time and in space by shutting them down. And again, we can do this in isolated brain tissue and also in the whole organism. And as I was saying, is this can tell us whether the activity of a certain kind of nerve cell is necessary. So Channelrhodopsin, by activating neurons, photostimulating them, we can determine whether it's sufficient. With the Halohodopsin, we can take the neurons out of the circuit and determine whether their activity is necessary. So between these two tools, logically we can determine whether neurons' activity is both necessary and sufficient. So we can really establish cause and effect in terms of the function of whatever system we're studying, the animal's behavior or the circuit's behavior. Okay, now again, in terms of translational stuff, what can we do with that? One example that comes to my mind, again in terms of human clinical therapies, epilepsy is due to an imbalance between excitation and inhibition. So the inhibitory processes in the brain are kind of impaired somehow, so that the excitatory processes take over, and it becomes kind of recurrent, out of control excitation. And this is a big problem for people who suffer from epilepsy. And so in principle, and again only in principle at this point, because Halohodopsin can control neuronal activity, shutting it down, you could imagine that this could someday become an important therapy for epilepsy. And just to complete, give credit where credit is due, this is important. This is Catarina Cunha. She's a student from Portugal. did the original work on Halohodopsin in their lab. And I told you about Guoping, and I told you about Karl. And Xuying also did some very important contributions on the project. So are there any questions about photo inhibition or Halohodopsin? Is that a question?
Can I make a stupid question?
No question is stupid.
Is there any possible relation between the color of the light that you are putting inside to the neurons and
I think he knows you.
and the using the light that comes into our eyes, through our eyes?
Well, that's not a stupid question because phototransduction in our retina also relies on rhodopsins, not channelrhodopsin and Halohodopsin, but plain old rhodopsin. And there are 3 different colors of rhodopsin. There's blue, green, and red. I think that's right. And so that's the 3 different colors of light in your color vision. And so the relationship between those is that they're both based on opsins, the kind of light-absorbing part of the channelrhodopsin and the regular rhodopsin. The details differ. The molecules, the channelrhodopsin is pretty different from the rhodopsin in terms of its function. The rhodopsin in our visual system, what it does is generate a second messenger cascade of a number of intracellular signals that kind of indirectly control the electrical activity of the receptors, photoreceptors in the neuron. Whereas the channelrhodopsin and Halohodopsin, the reason we like those is that it's direct. When they absorb the light, they immediately change the electrical properties of the nerve cells by virtue of being a channel or a pump. So, kind of in a general way, there's kind of molecular similarities, but in the details, they're quite different. In fact, the first thing that, not our lab, but Gero Wiesenbach's lab, who's at Oxford University now in England, he tried transplanting the whole, phototransduction cascade from a photoreceptor into another kind of nerve cell for photostimulation. It kind of worked, but it was not elegant. There were too many proteins required, too many genes, and too many intermediate steps. It was kind of messy. But channelrhodopsin and Halohodopsin, these are the real deal. It's the silver bullet. It's just one gene is all you need. So we like this much better.
Are there any implications for learning? So, for example, if you have a number of neutrons that was not activated before, and then if you learn a new skill and then you use the same neuron, would you be able to measure the quantity of certain neutrons that were
yes, and in fact, it's been shown again in the mouse brain. We're not talking about humans now. Kids are safe, don't worry. It's been shown that you can do that. You can use light-induced stimuli to replace, the normal stimulus, hearing a sound or getting some kind of visual stimulus through your eyes. You can replace those with just flashing the light to activate certain kind of neurons that are involved in learning and memory in the mouse brain. So yeah, you can really make learning a lot easier. Yeah. So again, we're not going to talk too much about the potential applications for humans, but you can. Doesn't take too much of a stretch of the imagination to imagine how valuable this could, in theory, be.
So George, just a last question. So regarding memory, what are you doing right now in your laboratory?
In our laboratory, right now we're working on fear conditioning in the amygdala.
Okay, using which technique?
Using the channelrhodopsin. So, yeah. So yeah, in particular, we've shown that behaviorally, this is done in collaboration with another lab in Korea. Behaviorally, we can replace auditory stimulus, the sound. So fear conditioning, the way you do it is you put the mice in a cage with an electrified floor. And so they don't like getting the shock on their feet. You wouldn't either, I promise. So what you can do is before you give the electrical shock to the feet, you can put a sound into the cage. Okay, and then they very quickly, you'd be impressed, very quickly learn when they hear that sound, they better run. actually what they do is freeze. Like, no, not again. And so we can completely replace the sound with the light flash. Yeah, very effective ones. So now what we're doing is trying to figure out which parts of, you know exactly, which parts of the auditory system are involved in providing the key information. Yeah, so that's one of the things we're doing. Okay, Maureen, how's our time? Time to quit? Time for more? I heard there's going to be wine. That's something I respond to very positively. But if you want to hear more, I've got about 10 more minutes. If you've had enough, I'm ready for wine.
Yeah, we can continue our conversation.
Okay, let's stop here. Let's see if there's any kind of emergent conclusion. Oh yeah, let's just go to the end here. We'll talk about that, we'll talk about that. So yeah, here's kind of the bottom line. In terms of this thing, optogenetic control of presynaptic activity, we're in very good shape. Channelrhodopsin and Halohodopsin, I showed you. They're wonderful. They really, truly work. In terms of this thing, optogenetic ways for monitoring postsynaptic responses, we're getting there, but it's not there yet. The thing that I didn't tell you about is this thing called clomelian, which allows us to look at the activity of inhibitory circuitry in the brain. And one of the big things that we're working on in Korea is developing optogenetic sensors for looking at excitatory activity, in particular, developing good optogenetic sensors of membrane potential. And then, so the bottom line here is that, we can, actually the bottom line is off the screen. That's telling somehow. But the basic concept is that it will improve throughput hugely. I told you that just using channelrhodopsin instead of traditional physiological techniques already improves throughput by 4 or 5 orders of magnitude. And by using these optogenetic technologies for imaging activity in the brain, we can improve that by another 1, 2, or 3 orders of magnitude. So suddenly this 100 trillion, 10 to the 14th, some unfathomably large number, suddenly starts to look a little bit doable. So we're very excited, very optimistic. It's a good time to be studying the brain. And, I'm glad that I could come here today to share our enthusiasm in this very exciting and important new technology with everyone. So thank you for your great questions. And I still want to find those kids and get some questions out of them too.